So welcome everyone. This is lecture number 9 of computational biology modeling methods using virtual tissues. Today, we will be talking about chemotaxis. We'll come back to the same topic we were discussing last week. And we will talk about Spml modeling to model subcellular processes and behaviors and how to incorporate these models into larger virtual tissue frameworks. I'll be going over the content of the class over the content related to the chemotaxis, and then we'll take a break, and then James will take from there to discuss subcellular modeling and SBML implementation in Compusal. Just a reminder, the class has been broadcasted in YouTube. So we will come back to some of the ideas we were talking about last time about the diffusion limited growth, what chemotaxis is and how it can be implemented as a linear response in Compusal. And then we will review the idea we spent some amount of time last week exploring how model specification parameters affect the velocity that cells move as they due to chemotaxis in response to gradients of chemical fields. We'll come back again to the idea that we went over rather quickly last time about how we could adapt the chemotaxis response such that the velocity of the cell does not increases exponentially as the gradient the cell is exposed to changes in an exponential way. And then we will explore a couple of examples of chemotaxis and how can they be used to model different types of self-movement and reproduce at least a model of bacterium microphage interactions. And then after that, James will take over and talk about the subcellular modeling Compusal in CC3D. Specifically with respect to the delta notch model and a cell cycle model. So before that, let's go over the room to get ideas of where everyone stands on their projects. As a reminder, the final version of the project, the first final versions of the projects are due on April 13. That's in 3 weeks. And the reason we want to have that buffer time is because we want everyone to be able to present the original versions of the project, get feedback, and also have plenty of time to make modifications before uploading your simulations as NanoHub tools. Remember that as part of your final presentation, you will need to include a YouTube video showing how to use the NanoHub tool that you develop as part of your model, that you deployed as a version of your model. Okay, those are reminders, but let's go around the room. The first one is Fanbo.
Yes. Can I share my screen? Yeah. So basically, for the last week task, I finished like doing all the documentations, trying to generate some like diagrams, trying to like structure my my project. Basically, I guess. So basically, these are the documentations, and I won't go in detail here. I only show this feature diagram. So basically, it represents all the functions and all the tricks I have implemented in this project. Basically, the client inside like the user interface. So they go to the Jupyter notebook and they can create their own user code and create their own mechanical object. By using their own code, they can use my widgets function to start, pause, and reset the image and do several like mouse interactions to move the images. And these responses are talking with the IPython kernel directly with the COMM interface. And these are the backend side, and it includes the like several widgets and also the threading. So the task for this week is like it basically concerned about like how to package all my code into a self-contained Python package, and also we have to make it like packaging inside the Mechanica, so the user can only call to install the Mechanica and call the Mechanica dot like Jupyter to call all the functions. The last thing is trying to make make a preparation of 3 versions of package for 3 kinds of users. So there are 3 kinds of users that may install our package. The first is only the desktop user. They will only use the desktop components and without installing like the Jupyter server side. So we will not include the Jupyter server, like the Jupyter notebook package for them. And the second is for the web end user. They will not install the package, but they will only go to the NanoHub in order to write their models and to run the models. So it's kind of a binary form for them. And the third one is for the server-side users. So typically for the NanoHub administrators who want to install like, our package in their server. And this should include like the Jupyter server-side component and also the mechanical components. Yeah, that's all for the task for this week.
Andy, do you have comments?
I think that looks fine, Andy. I mean, you're working, I know, with Fanbo. Do you have anything you want to discuss about how to go forward and what how we're going to this release?
Actually, this is very good. Yeah, this is all what Fanbo and I discussed a few days ago, and yeah, it's working. This is I think it's really good. One slight thing I do want to add is that what we're looking at is basically based on Fanbo's and I's discussion, sort of feedback thing. I'm adding a couple of events in the simulator itself, which will enable us to hook up Fanbo's code basically at runtime. And the advantage of that is that end users could run literally the exact same Python code on desktop and in Jupyter Notebook just by copying and pasting it. So it just the simulator figures out what environment that it is running in and then sets up the appropriate windowing system and communication mechanism with the other bits. So.
Well, I certainly think after our experiences running Compusal on NanoHub, having a Jupyter Notebook interface for the tool will be great. Not having to go through X11 would be very nice.
Yeah.
Andy, is this gonna be the first deployment of Mechanica on NanoHub, or is there is there like a previous version of it?
We basically have the existing version right now on NanoHub. It is in Jupyter notebooks, but it doesn't have. I mean, the displaying output and so forth is really clunky because it doesn't have Fanbo's interaction widget. But nonetheless, it's there and running on NanoHub Dev. And yeah, it's callable from like a Jupyter notebook and importing it, but it's super clunky of like actually getting output and displaying it there.
Right. We actually students might need your help at some point when we get to the point where they're ready to upload their tools to NanoHub.
Oh, great! Yeah, I mean, Giuliano and I are glad to help. So.
Thank you.
So I actually had a question that I heard in the conversation yesterday, which is, did they say that you could actually ask for 4 threads for a given application?
That's what I heard as well. Yeah.
So Josh, why don't we try multi-threading one of our demos today and see if it actually speeds it up?
Yeah. Yeah, I mean, I think, yeah, Paul yesterday said that each application can have 4 threads.
We could just try it. When we do our first demo, we can try it.
Yeah, you know what? I mean, I know with my code running on dev.
Let's not take the time for people now. But I just thought since you were talking about user experience, it occurred to me that I wanted to make that was something I wanted to put into this class to try out. If it works, great. If not, we've set the number of threads back to 1.
Yeah.
But I just wanted to check on that.
Yeah. The other.
Okay, great.
Yeah.
That's fine.
I just don't know how they restrict the number of threads, but okay.
Okay, Josh, go ahead, please.
So next in line is Jeffrey.
Okay, I have some slides. So first, this is an oscillating plate with a velocity boundary that's a function of the time step, so it oscillates back and forth. You can see here, and as I was adjusting the parameters, I realized that I think the issue with the difference between this analytic solution and the one I'm plotting is that the top velocity is being mapped down to the bottom here, because you can see here the velocity is basically 0, but it's showing that it's very high. So I think once I figure out why it's doing that, the analytic solution and what I have plotted will fit a lot more closely than it is now. And then last night, Andy updated Mechanica, and I was working on getting the convection cells going with this catalyst here in the bottom. So this is the XZ plane and YZ plane, the side, and then here's the top. And you can see this is just kind of a rough first version at it, but the convection cells are more or less forming. And I also have the actual simulation of it. It's a little hard to see because I have it working. This is the catalyst. Because the boundary conditions are all no slip. So on the outside it looks still, but if you look closely, you can see here the red chemical filled cells, once they've reacted, are lighter and are moving up, and it's a little easier to see, I guess. Here on the top you can see it's kind of bubbling up in the middle. Yeah, you can see the red cells going up kind of. And so this is more or less the end flow result. In the paper, they, I think, add other objects that the flow moves, so that can be something I look at again. But I was glad to see that this is working and the convection cells are forming as the chemicals react.
Oh, okay, so this is just where you hooked up that vertical force to a chemical on the object itself, right?
Yeah.
Cool. Okay, and it's forming the convection cells. Excellent. Okay.
It's effectively like chemotaxis if they were biological cells.
Mm-hmm.
So in terms of the rendering that we're seeing in here, is this rendering that Jeffrey, are you doing the rendering? Is this rendering Andy? You're supplying this rendering image, the video?
Jeffrey, I assume you did like a screen capture or something?
Yeah, this is a screen capture.
No, I'm just wondering because, I mean, for this, it would be nice to be able to make some fraction of the particles transparent. So you could see a slice through the image.
Mm-hmm. Yeah, and then what I'll do, what I'll try and get to probably next day or so, is in the style object I could add a slice plane,
a cut plane, or
yeah.
I mean, of course, there are all these possibilities. You might want to take out a quadrant or something, but at least to begin with, a cut plane would be nice.
Can
we look back at number 3, slide 3 for a second?
Yeah.
So one thing I'm wondering about a bit is, I mean, the streamlines generally make sense, but they're, I mean, if I look at the top right one, for example, I see that vortex that's forming in the convective cell. But then if I look down lower, sort of at the bottom half in the Z direction, there's a lot, it's pretty noisy. And I'm wondering where that noise is coming from. Is that because of the volume over which you're averaging? So there aren't enough particles inside of the averaging volume to be smooth? Or is this a moving average? In other words, you're drawing a box and you're calculating the mean velocity of all the particles in the box. How are you calculating the streamlines?
So this is a slice in the middle of the domain, like the 3. So it's just that, and then each point, or the whole simulation, is divided into grids to get numpy arrays. So it's like putting each particle into a little box, and then you average the number of particles in that box.
So you're averaging the direction of movement of the velocity, the average velocity of the particles in the box, in the XY, in the direction of the plane, within the direction of the plane.
Yeah.
Okay. And then you're using some NumPy function to calculate streamlines based on the individual particle velocity, individual vectors in the boxes.
Yeah, it's the stream plot function.
I'm just wondering why it's so noisy.
It could also be because just the parameters aren't ideal. I just got roughly the kind of flow pattern that I wanted, and didn't really. It could be there's a random force. It could be too strong.
I think you did this pretty early on, so it probably maybe it hasn't settled down to a steady state.
Yeah, that's also a problem.
I mean, one thing you could do that might help, given how those stream function calculators work, is to take the slice and increase the resolution. So suppose, I don't know how many boxes you're using from left to right. It'll maybe 20 boxes across.
Yeah.
How many domains? Okay. So
yeah.
so instead of using 20 boxes, do 200 boxes, and then have each box have overlapping boxes that move just a little bit, like this.
Okay.
So that your NumPy array that you're calculating the streamlines from is actually 10 times the resolution or 100 times the resolution of the actual NumPy array.
Okay.
And that'll smooth the calculation.
Okay.
And then when it comes to try to rebuild the streamlines, it may do better.
Okay.
Because there's no universally good way of calculating streamlines from a bunch of vectors.
Mm-hmm.
And so there's always a bit of a game to play with that. Actually, this is something that LF is a great specialist in, is how to do those kind of reconstructions.
Okay.
Because he, in his diffusion tensor plots for neural net for brains, the diffusion tensor gives you a vector at each point in your MRI image in 3D, and then you want to try to calculate what that means in terms of some imagined underlying connectivity of the neuronal architecture. And so, but the signal to noise in those MRIs is extremely poor, and the spatial resolution is also poor. And so when you see these beautiful plot diagrams of neuronal connectivity, if you saw the underlying data sets those are inferred from, they're the underlying data sets are very noisy and very sparse. And so there's a lot of work to go from these sort of particle-like methods where you get a PIV, particle imaging velocimetry, to try to go backward to get the streamlines. And so it may be that some of this may be that you're not in steady state. Some of it may be that the number of particles is not enormous. It's a lot from the point of view of doing the calculations, not so many from the point of getting smooth velocity field. But some of it may be because of the way you're doing the velocity calculation in the plot.
Okay.
And so I'd encourage you to try playing with the way you calculate the streamlines and see if you can get a smoother one. Not so much for us. This is fine, but if you're going to publish it, and you don't want to be dishonest, you don't want to pretend that you're getting better streamlines than you are. But I think just from a lot of those short streamlines that are sort of random or colliding with each other, streamlines aren't allowed to collide.
Yeah.
And so that suggests that it's failing to be able to do the inference.
Okay.
And that usually is because the boxes are too far apart. And so the angle change between the neighboring boxes is too big. And so if you do a window average with a finer resolution than the one you started with, it'll slow down the plot, but it should fix that problem.
Okay.
And I'm happy to play with it offline if you want to meet about that. I'm sure Andy knows how to do this too.
Okay.
And that may give you slightly nicer plots.
Okay. Yeah, I do have the resolution kind of low right now, just so that the plots. So for those videos, the plots aren't taking forever to update. So that is probably another thing to look at. Yeah.
Well, if you actually store the position of velocity of each particle, then you can do the image calculation offline.
Yeah, I've been having an issue with the FFmpg layer. I'm still trying to figure out how to get that to work so I can just plot the data from a file and make a movie. But
I mean, the base results are very good. I'm just thinking, I think about ways of displaying them that might be a little bit clearer.
Okay. Yeah.
One thing I maybe could add is add something to the style where basically doing streamlines of basically as picking a number of objects and actually having them trace their trajectories as they go through the domain.
Mhm.
So Eulerian versus Lagrangian approach. Yeah, well, that's reasonable. Again, there's no perfect way to do streamline calculations. But I think, and when you do a slice, you're also losing a lot of information because particles come in and out of plane.
Yeah.
So there are a bunch of things we could do. So it's probably worth actually saving the position and velocity of all your particles, even though it's a big file,
Okay.
so that you can then play with some reconstruct. Because they're very sophisticated techniques available for reconstructing streamlines for fluids.
Okay.
And so we could look at what's out there for doing that. Again, that doesn't change the fact that it's working, which is the main thing. It's more a question of preparing this for publication, how to present it most effectively.
And something else. So Jeffrey, I assume what you're doing is every so many time steps, you're calling the grid, which the grid function, which basically gives you back that three-dimensional vector of particles sorted in that grid. That's a snapshot in time. Maybe if you did that, say every, I don't know, 2-3 time steps, and then average those over time.
Okay. Yeah.
Yes. So basically, what I'm saying is, right now we only have spatial averaging, right? We don't have temporal averaging.
Yeah.
And the passive trace idea that you had, Andy, is also a good one, where you actually plot the position of each particle versus time over some period of time.
Yeah.
Again, the thing you have to worry about is if they go out of plane, you have to have some way of handling that.
Yeah.
But
yeah, I mean, on the rendering side, it's not that big of a deal to add to make an object a tracer, and then basically calculate and basically save that as a list of line segments, and then
exactly.
Yeah.
So maybe you can work with Jeffrey a little bit on better ways, different ways of representing those kind of images. Yeah. Again, this is certainly clear about what you've got, but
it can be better. Yeah.
Okay, so maybe that's something you could put on your agenda for next week, would be thinking about displays.
Okay, yeah.
Okay, great. Okay, thank you.
Hey, I have Callie next.
Did you say me?
Yep.
Okay, I thought I heard Calvin for some reason. I'm like, there's no Calvin in the class.
Callie.
Well, it's funny. I mean, if you look at the transcript, it got Calvin and Kelvin. It's trying to guess, and it doesn't recognize your name, I guess, and so.
Yep. All right. So my update for today. My goals for this week were to work on to finish the cell initialization, which is not quite done yet, but I am playing with graphs as seen below. Add the ammonia field to the model, which impacts cyclic AMP dynamics. So basically add another chemical field to my model, which impacts cell excitability, diffusion, and production of ammonia, which I accomplished, and add chemotaxis towards areas of higher cyclic AMP concentration, which was also accomplished. So threefold update. Here is my progress so far on the cell initialization. On the left, we have the initial condition. This is what we're going for. And on the right, we have a series of graphs that are beginning to represent this initial structure. So we have the different cell types. There are some others, I think, in this graph that aren't necessarily visible. For this exercise, I was just kind of going for the major structures. So we have some green, I think these are PSTA cells here, so pre-stalk cells. I think red is PSTO, another type of pre-stalk cells. And then we have our pathfinder cells up top with the blue, the dark blue, stalk cells in like the lighter blue, the cyan. I think the pathfinder cells are this purple. Or one of these were pre-spore. The pre-spore cells might be the blue instead. I'd have to go back and look at the paper. But anyway, so on the...
I would have thought the red were the pre-spore, but maybe not.
I'd have to go back and check that. But anyway, so on the right, we kind of have the major chunks sectioned off so that the colors don't line up with what we'd expect, what the initial condition has. But we have our lower bottom baseline, our red cells, and then 3 other graphs for the blue, the dark blue, light blue, and purple cells are kind of not very visible in this diagram. So that's just 1, 2, 3, green, purple, black. This is an imperfect diagram, and I'm unsure how to, I'm not sure when to say whether a certain initialization is good enough here, because these are qualitative results. So it's not, I don't think we have access to like a spot by spot, here's the initial condition of every single cell or the initial location.
I would say as long as the number of cells is approximately right, within 5% or 10% of what they started with.
All right.
And the basic geometry is all right. The details are not going to matter. If the chemotaxis works and you actually get the thing to pump up and make a stalk, that'll be fine. Okay. Because in his later work, he actually showed that there are different strains of dicty with mutations, where the chemotaxis is different, where the signaling is different, and those give rise to different morphologies of the stalk. In addition, the simulation makes it look like you always get exactly the same thing, but in fact, you can have a big variety of difference in the number of cells in a mound that forms a stalk. So you have a little teeny one that makes a teeny one, a stalk, or a big one that makes a big one. It can be irregular in shape and still works pretty well. There also are variations so that in some mutants, the fraction of cells that become stalk is bigger. In some mutants, the fraction of cells that become spore are bigger. And so as long as you have something that's approximately there, that's fine. If you can replicate the base result, you can then check on how as you change those parameters, you can check on how the morphology of the solution changes. So I think that in terms of the geometry, you're just fine with what you've got.
Okay. So that's the geometry. Now going on to the coding. So as I kind of mentioned last week, I had a mini chemotaxis model, so like a little toy model with my group of 9 cells. And then I also have what I'm calling my mini project preliminary model, which contains all the actual cell types that are included. So in the mini chemotaxis model, I implemented the updated cyclic AMP model in the fruiting body paper by Murray, which accounts for the impact of ammonia. And that impact is accounted for by having one of the excitability variable in the model be dependent on the concentration of ammonia in the cell. With that, okay, excellent. Went to the next slide, one I shouldn't have. Also added ammonia as a chemical field in the model and added the production and diffusion. Production I added as a secretion from each cell, and then diffusion is diffusion. And then because chemotaxis with cyclic AMP is in this model is determined by the conditions of cyclic AMP concentration and cell refractoriness at that time, I implemented the chemotaxis in Python instead of in the XML file, and having every time we check for the conditions. And then if the conditions are met, we update our Lambda so then chemotaxis can occur. If not, it's at 0. And then I went and began transferring this code over to my mini project preliminary model, but then replacing my basic oscillatory and relay cell types with the pre-stalk A, pre-stalk O, and pre-spore cells, which are all implicated by these, by the cyclic AMP and ammonia. So with that, I could show some code now if that's something you'd want to see.
I mean, I'm impressed in the sense. My main worry about this, about implementing Stan's model, was that Stan didn't separate what was going on inside the cells as a chemical reaction inside the cells from the diffusion equation. And so if you're able to do that separation so that the diffusion and the secretion absorption work, and then that cell cycle within the cells work, I think everything else. I'm not saying there are no mines to step on, but that's the main. I thought it would probably be the main difficulty here. So let's take a look at what you've got. Actually, you're going to be basically you're teaching this class because you're covering all of the things that Josh and I were planning on covering, although in a little more detail than we were going to do it. But.
All right.
So everybody can watch this, and then maybe the things that as a teaser for what we're going to cover that in class.
All right. So the way I had originally implemented this anemone string was using a more basic cell excitability parameter A of T, which was included in the earlier paper by Murray, Panthalov, and Algaweg. I still don't know how to say that. But in this later paper, the one that I'm primarily basing my model off of, there's an altered version of this parameter called A of Tn, and N is kind of standing for like NH3 ammonia here. So this is like the this is the cell excitability that accounts for the ammonia concentration, and it uses the old parameter A of T and a new parameter B of T to make that calculation. That's also dependent on the concentration of ammonia N here. So that's the update to the anemone string. So kind of the same thing as before, the anemone model gets added to all the cells. We adjust the cyclic AMP and ammonia concentrations based on what the cell is seeing. There may be an argument, I know Dr. Zico was saying, instead of doing amount seen by cell, it's amount, we could do like the amount of ammonia at the center of mass of the cell. And then
Actually, I have one quick comment. If you go back to the antimony for just a second. So you're updating the value of N, so you're writing the value of N into the cell, is that right? So
yes.
You're reading the value. Okay, so then you're going to have the usual issue, which is A underscore Tn should probably have a colon equals, because otherwise it'll only read it once.
Got it. That's a good catch.
So
Here too.
There's always that peculiarity when you use the. I think, let's see. So Fanbo probably hasn't worked with antimony, and Drew, I guess, hasn't either. If I don't know what's been happening, Drew's been logging in and disappearing, so I hope the network's all right. But in any case, everyone else will remember there's this peculiarity in antimony that it's parsed once at the beginning of the simulation, and then if you don't tell it you have to update, it doesn't. So that's the one thing I would notice on that. Otherwise, that looks reasonable.
So they updated some of these parameters based on what the new paper said. Update C and N based on what the cell is seeing. if the cell is a relay cell, remember this is the mini chemotaxis model, not the actual model. So in the when we have the actual code, it'll say if cell type is pre-stalk O or pre-spore. We change the A of T and B of T variables. I decided to start a dictionary of the excitability terms. And then we add a chemotaxis, and we add chemotaxis to that cell in Python instead of in the XML. And then in every step, we're going to get the...
There's a typo in line 88.
Where?
Add chemotaxis to
Oh, Lord. I changed them in both places. All right, so after we fix our typos, then we get the fields for cyclic AMP and ammonia. We find the amount that the cell sees. Then we're going to add that chemotaxis. And then if the conditions are right, if the cyclic AMP concentration is greater than 0.05 and our refractoriness is less than 0.2, we're going to set lambda, our chemotaxis lambda to 200 because that's what the paper says. And we're going to have chemotaxis. But if not, then we set it to 0 and we have no chemotaxis. And then, yeah, so then that's the mini chemotaxis model. It's analogous in my actual model, except instead of saying oscillatory and relay cells, we're saying pre-stalk A, pre-stalk O, and pre-spore. So it's a little bit longer. And then in the XML, we've added, let me go to the mini chemotaxis one. In addition to the cyclic AMP field, we have added ammonia, and the ammonia in this model is done globally, not by cell type. So we have our global diffusion constant, which is 15. And then they didn't give a decay constant in the paper, but they did give a production for ammonia. And for all cell types, I think it's that this is involved, it's 0.001. And I think that covers both of my updates with the chemotaxis and the ammonia.
So one question which you may have to, I hope they're clear about, is where the diffusion happens. Is the diffusion only in the cells and excluded from the medium, or does the chemicals diffuse in the medium as well?
Yeah, so for cyclic AMP, it's cells only, but ammonia can diffuse in the air and the soil, I think.
Great. Okay. Because that's my guess is that it'll work either way, but probably the parameters will be different, right?
Mm-hmm.
So have you gotten, how close are you to having something you could try to run and see whether it does something?
Depends on when I get that initialization done. I mean, I don't like it. I could probably run something maybe without the initialization done, maybe in the mini chemotaxis model. But as far as the full diffuse discordium model, we would need the initialization to have been accomplished first.
I think it might be worth trying to do the kind of simulation I showed the other day, which is throw a bunch of cells down on 2D, and some relaying ones and some oscillating ones, and see what they do. So just a simple geometry where you put them down on a grid with some spacing, and see how they respond.
Okay.
Because that way you could test all the pieces of your model, even if the geometry isn't quite right. Because the way during culmination, they're also differentiating and becoming stalk, and the stalk cells are dead and don't do much.
Right.
But the chemotaxis part of the model is the same one that they do during aggregation. And so you should be able to see the cells come together to aggregate and form streams. And of course, Josh showed us last week, some of the movies, some of the classic movies of Dicty doing their thing.
Yeah.
And so but this is great. I hope we can get this to work. TJ, did you have any comments you wanted to make about?
No, I mean, making great progress. The one bit of work I've not seen since we last talked was the progress on the initialization. And I think that approach, the approach that's been implemented is the right way to go. Probably the most difficult part of that will be actually seeding within the domains that are defined by those curves. I mean, I would think that if we could get cell numbers that were within 5% of what is described in the paper, that would be acceptable. Because they do actually state exactly how many cells of each type they use, which is really nice. But we'll have to mess with the algorithm a little bit to correctly seed those domains. But no, great progress. This looks great.
One thing I will add, I played around a little bit with this model a while back, and Murray's equations are set up such that a lot of the chemical concentrations are negative. So it's just something to be aware of.
Does concentrations go negative?
Yeah, a lot of them are continuously negative, and some of them fluctuate between positive and negative.
Okay.
And I mean, I could be wrong. Maybe I typed them in wrong or something. But that's what in my case, that's what I was always saying. But it might be worthwhile to type up his reaction equations and simulate them by themselves in Tellurium or in Mathematica or something, just so you can see what the reactions end up doing before you stick it in a spatial model.
Yeah, so Callie prototyped that a few weeks ago, and we unless I missed something, we didn't observe that in those prototypes. There may be some detail with the ammonia. The cyclic AMP is implemented a little strangely because, as Callie mentioned, there's no decay uptake described. And at least for the cyclic AMP, they manage that by imposing a constant concentration of 0 in the medium. So just have production inside the cells and then a constant boundary condition of 0, basically. For the ammonia, it's a little less clear, and I don't think that we're confident in any particular solution on that issue yet. But yeah, negative values is not something that I think has really come up on our radar.
Yeah, so just like I said, that's what I observed. I might have made a mistake. I might have typed them in wrong. I mean, but just something to be aware of. So okay.
Okay, cool. As James says, as James said earlier, a lot of the stuff that Callie was showing is related to the topics that we will be covering today in class. So I hope everyone was able to get a little bit of a teaser of those topics.
Right.
I still have.
I guess. Hold on one sec. I just wanted to say, I put in all of Stan's papers are available individually, but it's convenient. I put into the chat the link to his PhD thesis. And the thesis is nice because the papers are all in sequence. And so having them aggregated that way may be helpful. And one thing that we haven't tried, which maybe is more for TJ than Callie, is he has one of the early chapters of the thesis is doing purely the PDEs. The PDE spiral wave generation. And that should be, I don't know because it's piecewise linear whether our reaction diffusion solver can deal with the PDEs that way. But it's something to think about. And so you might want to, if you haven't looked at the thesis as presented, it might be worth walking through the thesis and seeing if there's sort of hidden material, for example, let me just, actually, can I screen share for a second? For example, this is his model of aggregation, and so if we could replicate that aggregation result, that might be interesting. So,
Dr. Glazier, could you send this in the chat again? I don't think I see it.
Oh, I see what happened. Okay, sorry. Usually it goes the opposite. What happens usually in the chat, somebody intends to send a private message and they accidentally send it to everybody in the room. In this case, Andy had sent me a private message and that automatically sets it so that when I respond without noticing it, it sends it back to him only. So there you go. Did you get it now?
Yeah, I have it now.
Okay. And I should say, I mean, I don't know if anybody who's ever going to write a PhD, I would say that Stan's thesis is one of the most exemplary pieces of work I've ever seen as a PhD thesis. Now the Dutch theses are mostly published papers, but there's a coherence to the way he put that together that's quite remarkable. The other thing that the Dutch did, which is not maybe as much of an issue in these days when everything is electronic, but used to be a big deal, used to cost money, was that the Dutch required you to publish your thesis. And so in those days, that meant you paid some small publishing company to basically print 100 copies of your thesis, but you got in those days, you got an ISBN number for it. And so it did count as a book. And I think that was always a good thing from the point of view of the students to have that on their CVs and otherwise. But in any case, that Stan's thesis is some beautiful, beautiful work. Almost every chapter in it would be a great topic for replication, and he writes beautifully as well. So his thesis advisor, Pauline Hageveg, was a remarkable scientist, and Sasha Panfilov also great team in Utrecht. Okay, so I just wanted to show that image. I'll stop sharing. I guess I already stopped sharing, didn't I? So back to you.
Yeah, so we have next in line is Tyler.
Yeah, could I share my screen really fast? Okay, so I've constructed a flow diagram to outline the overall outlook of my project. So, so far I have nearly every element here implemented to some fashion. Some of it needs tweak still, mostly the IL2 field, which, as you can see, lies at the center of this entire flow diagram pretty much. But as I described before, we have naive cells that when they come in contact with APC, they become pre-activated. And once pre-activated, they will secrete IL-2. And once the IL-2 threshold for pre-activated cells is reached, they become activated. And from here, once the TBET threshold is reached for activated cells, they will become effector. And pre-activated, activated, and effector cells all undergo apoptosis, caspase-mediated apoptosis. And this caspase is also catalyzed by Phos and Phos-L interaction when activated and effector cells come into contact with each other. And that is still part of my project. That is one of the, excuse me, that is still an element that I need to implement. So for this week, what I wanted to do was implement cell motility for all cell types, which I've successfully done. I wanted to implement the caspase-mediated apoptosis for those 3 cell types. And I still need to work on differentiating for the Il2 differentiation and Tibet differentiation. That is, as you can see here, I have screenshots. The secretion of IL-2 from my pre-activated cells here in green, that is working. However, I still need to work on calculating the threshold values so that they differentiate appropriately. Right now I have it so that I have it in fragments right now. And when all of it is activated, my naive cells for some reason die immediately, or they immediately turn into effector cells, which is something I need to work on. But every element can be seen in my model right now. But like I said before, I pretty much just need to tweak everything to get it all working in line. And that's what I want to prioritize the IL-2 diffusion field for this week, because that will allow me to see the differentiation for at least pre-activated and activated cell types. And then from there, the Tibet and caspase-mediated apoptosis should be working fine.
Can we come back to that slide 4 for just a second? So I don't know what, I mean, the absolute numbers don't mean anything, probably. But if I look at that IL-2 plot, the maximum value is 2 times 10 to the minus 11th.
Right. The units that they have in the paper is in molarity, and I'm working on converting it to Compusal units, and finding the value in order to get the field to work properly has been a little difficult.
Well, usually these so small numbers are expected because you're usually talking about nanomolar or even picomolar concentrations for cytokines. So you might want to re-express things in micromolar units or nanomolar units.
Okay.
Just so the numerical values are more reasonable. In other words, multiply by 10 to the 9th, 10 to the 12th for everything, picomolar. So that you have to then convert your units, but I mean, it doesn't matter. You can always multiply by 10 to the 11th when you set your threshold. But it's sometimes convenient to try to keep your numbers sort of a reasonable, not too far away from 1. But and so what's the rule again for what kind of rule you have for? So the role of IL-2 is. Can you walk us through the diagram again? Because that was.
Yeah, that was a little fast. IL-2 is secreted by pre-activated, activated, and effector.
Mm-hmm.
But IL-2 is the driving force for pre-activated to become activated.
Okay, so there's going to be some rate equation for pre-activated becomes activated, which will be a function of the IL-2 concentration.
Correct. Okay. And it's that activation, it's this, the threshold value for pre-activated to activated that I'm not sure why it's not working, but right now the code works so that when the APC comes in contact with naive, it becomes pre-activated. And then if the APC comes in contact with the pre-activated cell again, it becomes activated, which isn't correct. And that's what Dr. Isigo and I have been working on for this week.
Okay. And so the pre-activated cell reads the IL-2 concentration in its neighborhood?
Yes. Okay.
And you've got something that reports what it's seeing, so you know what level of IL-2 the cell is seeing?
That's correct.
Okay. And is that a deterministic transition, or is it a rate law where you say the probability of going pre-activated to activated depends on the concentration?
It depends on the concentration.
But is it a fixed threshold where it says if the concentration is above X, you do it, or is it that the probability of changing depends on the concentration?
It is a fixed threshold.
Okay, because that may be a little fussier. Having a rate law that's sort of a hill function of the concentration may be better, may be easier. If the concentration is bouncing around, then having the transition deterministically on a particular threshold can be an issue. Again, I'd be happy to meet and talk that one over at some point.
Sure. Yep. But yeah, that's my main focus is correcting the IL-2 field and getting that working properly. And that's my main priority for this week. And then everything else should fall in line with that. And that's pretty much all my updates for this week.
Okay, Josh.
Okay, what I'll do is I'm gonna go over the stuff that we covered last week a little bit quick, so we can get to the in-class exercise in a short amount of time. So the first thing we talked about last week was diffusion limited growth and limited growth, and in that case we talked about how cell growth and proliferation is usually controlled by the availability of resources in the cell's environment, and that we can model that by modifying the rates of growth, making it a function of how much resources that are available to the cell, and the cell is able to take up from the environment. And we looked at a couple of different functional forms for those for implementing those or modeling diffusion limited growth. And one of the things we saw is that the morphology of the cellular blobs, if you want to think about this as tumors, the morphology of tumors is going to be dependent on the specific form that we choose for our the specific mathematical form we choose for limiting growth. In this particular case, since cells are basically penalized for having no exposure to the resource, that is going to drive the system to maximize the contact surface between the source of the metabolic resource and the surface of the tumor. And that's why you get this particular type of branching. And by playing with the diffusion rate, the diffusion coefficient, or the diffusion length, and the uptake rates, you can arrive at different morphologies. And there is at least one paper using Compusal that explores and casts this morphology in terms of how invasive the tumor is with respect to its microenvironment. So that is just something to keep in mind. Then we talked about cell movement, and we talked about how cells move along different types of gradients, and we can summarize, or we can model all of these type of gradient-driven movements as chemotaxis. And chemotaxis refers to the movement of cells along chemical gradients, and these chemical gradients can be due to gradients of oxygen or other metabolic resources such as glucose. It can be away from different toxic molecules, or it can be in response to certain signaling molecules. And that's particularly true in the case, for example, of immune cells responding to a particular signal because of a specific lesion in the tissue. And then we talked about how chemotaxis is an important mechanisms for cells to move from areas of low resources to high resources or along the signals, and we showed these videos of this totilium, and the model that Callie was talking about is a model of this totilium development, especially specifically in the phase where it develops. It develops into this bud. But in these videos, you can see how the cells move when pipated with cyclic AMP into the media. Cells move and chemotax towards the areas where this cyclic AMP is being provided as they detect the gradient and detect what are the areas that have higher concentration of the molecule. And when we talked about how if we want to model chemotaxis, the simplest way we can do it is to generate a force that acts on cells, and that a force that is linear on the gradient of the chemical field that the cell is exposed to. And the idea here is that then the important idea here is that the response will be linear on the gradient, which means that if the gradient is linear, the response or the speed will be linear, will increase linearly, or it will remain constant. It depends, right? But if the gradient is increasing exponentially, the speed at which cells move due to that gradient will also increase exponentially, and that might represent certain problems, both in computationally, because in the specific case of Compusal, that cells might disintegrate due to experiencing forces that are too high, or there might be biologically unrealistic, because in reality cells do not respond entirely linearly to gradients, especially as those gradients become steeper and steeper. But we'll come back to that later. We mentioned that the way that this chemotaxis is implemented in Compusal is by adding another effective energy term that we call chemotaxis energy, and that the chemotaxis energy is linear in the gradient, and it's proportional to this. Is the proportionality constant is this lambda chemotaxis, and the importance about this lambda chemotaxis is that it's going to control both the strength of the signal of the response and the signal or direction of it. So if chemotaxis is negative, it means that the cell will have the chemical field will act as a chemo repulsion, and the cell will move away from areas of high concentration. If the lambda chemotaxis is positive, the chemical field acts as a chemoattractant, and the cell moves along or from along the gradient up to higher concentrations of the chemical field. We showed that there's two ways of specifying chemotaxis in Compusal. One is you doing it on XML, and the advantage of doing it in XML is it's quite simple, but it gives you very little control over the chemotaxis. So the lambda chemotaxis cannot be reset dynamically through the simulation. Then we created the simulation where we initialize a cell in a linear gradient. And what we saw was that the speed of the cell was constant as the cell moved through this linear gradient. and we played a little bit with different values of the lambda chemotaxis. And in this particular example, what we saw was that as the lambda chemotaxis increases, the cells moves faster. And as the lambda chemotaxis decreases, the cell moves slower, but also the effect of the effect of the random fluctuations of the cell membrane are much more visible than in the cases where the lambda chemotaxis is higher. This means that when the lambda chemotaxis is smaller, the combination of movements due to random fluctuations and chemotaxis are on par, or very closely or closer to each other than in the case where lambda chemotaxis is much higher, where the cell movement is almost entirely driven by the chemotaxis. Then we also talked about how we can specify the lambda chemotaxis in Python, and this gives us much more control over how to set the lambda chemotaxis dynamically. And we exploited this ability to change the chemotaxis of the cell such that we could plot, we could create a plot of the velocity of the cell as a function of the chemotaxis, right? And in this particular simulation, what we did was that, I'm gonna, hold on one second, I'm gonna share my full screen. In this particular simulation, what we did is that we started with a cell in the linear gradient, and then we specified the conditions such that the cell, as the cell reaches the border of the simulation, we switch the direction, the sign of the lambda chemotaxis. So we'll make the cell move on the opposite direction, and then we increase the lambda chemotaxis at a constant rate. And what we saw is that as we increase that lambda chemotaxis, the velocity of the cell increases. But there is a point at which the speed of the cell saturates, and even though we keep increasing the lambda chemotaxis, we do not see a corresponding increase in the speed of the cell. And the reason for that is because the way the algorithm is implemented in Compusal, a pixel can be updated just once per Monte Carlo step. So if you have at a boundary of the cell, the boundary of the cell can just move one pixel at a time per Monte Carlo step. There's no way for the boundary, for example, to move 2 pixels in one Monte Carlo step. So the first exercise we will do is, now we did this for the lambda chemotaxis, but we can use the same code to see what other parameters do to how other parameters affect the chemotactic response. And I'll show one first. I'm gonna call the polling because we will keep asking people to show their codes. I'll show one first, and then I'll ask people to show theirs as well. So the first thing we'll test is how the cell size or the cell volume affects the chemotaxis. So the idea in this case will be the same. We'll have the cell move in one direction, then we will change the lambda chemotaxis, but instead of increasing the lambda chemotaxis, what we will do is that we will increase the target volume of the cell. And then we can plot the velocity, instead of as a function of lambda chemotaxis, we can plot it as a function of the cell volume. The first thing to note here, and I think this was part of the homework, was to choose a reasonable value for that lambda chemotaxis that we're not gonna increase. And we shouldn't, and we can see that in this range around lambda chemotaxis up to 1,000, we do not see a very good response of the chemotaxis. It's very noisy up there due to the gradient that we have. So lambda chemotaxis should probably be higher than 1,000. And we see here that over 10,000, the velocity is starting to saturate. So that might actually not be a good starting value for the lambda chemotaxis. So what I'll do is I'll start with a like around here, like around 5,000 for a initial lambda chemotaxis. So we can if you guys go to Canvas, there is the lambda chemotaxis, lambda chemo in lecture 8, in demos lecture 8, that is lambda chemo, and this is just this file, this this script that you guys are seeing here. But now what we want to do is that we want to be able to increase the cell volume when we change the direction of the lambda. So the first thing to do is to comment out the volume parameter specification in the XML, because we want to be able to modify it in the Python stepboards. James, by the way, I did try increasing the number of cores this is being running on, and I did notice a speed up.
So in that case, we should just default to using 4 cores.
Yeah, I did notice it. I'm not. It's in lecture 8 demos. It's called Lambda Chemo. Good. So yeah, I did notice I did notice a little bit of a speed up there. So yeah, we'll comment out the lambda chemotaxis, the volume specification in the XML, I'm sorry, and then we'll come here to the Python stepable. And then, as I said, we want to specify a lambda chemo that is reasonable within that linear range. So we'll set it to 5,000. And all we need to change now is, well, first we need to also specify, because we have deleted it in the XML, we need to specify a initial lambda volume, target volume, and lambda volume for the cell. And then what we will do next is that in the step function, in this condition here, 41, what it's doing is that it's measuring whether the cell is close to the right border or close to the left border, and then changing the lambda chemotaxis when the cell is near that border. But it's also because with the lambda chemotaxis, we don't know how fast the cell is moving at the beginning. We did not want to wait too long to update the lambda. So we also added an additional condition, and that condition was that at least 1,000 Monte Carlo step has passed. So even though the cell hadn't reached the border when the lambda chemotaxis was small, we still changed the lambda to be able to measure and increase it, because we were not getting a lot of movement from the cell. We'll keep that here, it doesn't hurt. But then what we want first is that when any of these conditions is met, the first thing we want to change the direction of the cell. So we do that by multiplying the lambda chemotaxis by -1. And then we also want to increase the target volume of the cell. By, let's say, 1.1. Let's make sure that everything is fine. So basically I added the target volume and lambda volume. I specify what the initial lambda chemotaxis will be. And then instead of increasing lambda, I'm increasing the target volume, but then I am still changing the direction of lambda. And then what I need, and then for the plotting, which is important, in the original plot, I was plotting the value of lambda and the velocity or the speed. But now what I really want to really want to plot is the cell that target volume. So now, as what we will see is, instead of velocity against target volume, is we're gonna see velocity against lambda chemotaxis. We're gonna see velocity against target volume. Okay, let's see. Okay, so now we can play the simulation. And we see that the cell is moving.
Is everybody able to do the upload of that file? Because I'm actually having some trouble getting it.
I'm trying to do it through the NanoHub Jupyter Notebook tool so then I can open it in Compusal.
Right, is Jupyter Notebook working? Because I keep getting a 404.
It's working for me. I'm just trying to figure out like if I'm able to upload this as a folder instead of individual files, or if I need to upload it as files and put it into a folder.
I think it should be able to be loaded as a zip and then use Twit to unzip it. But or does this? I don't remember. TJ, is the current version of Compusal? TJ isn't on it. TJ signed up. I was going to ask: do we have to unzip it by hand, or does
you just open it on Twit? You can just open it on Twit.
It does, yeah. Because earlier versions of Compusal didn't have that, but it should work. I mean, right. but I'm having tried for some reason. I can't launch Jupyter Notebook to do the upload. So give me a. So I'm the one who's holding things up, but we should make sure everybody's able to.
Right. So Callie, were you able to load it up?
Yeah, I'm in the process of doing it now. I'm just adding folders and then uploading each file and putting in the folders. I know there are more efficient ways to do this, but it'll get done.
That's fine. What about the other people? Have the other people been able to do so, to run it? What are you guys seeing?
So I have trouble with like running. So when I hit the run, like the cell just disappear. Like
Oh, so because I think the version I uploaded on, just make sure that the version because I was realizing earlier that the version I uploaded had the lambda volume here, this information was commented out. So make sure that that information is indeed commented out.
Yes, I have comment like the volume constraint.
Right. So right. So the right, but just the parameter specification, and then also make sure that you specify here when you create the cell.
Yeah, I think all things is the same as yours. And I can share the screen if like.
Yeah, does. Yeah.
Okay, thank you.
Yeah, no problem. Let me stop sharing it. You can share now.
Yeah, so basically I think all things is the same as yours. So here I come the volume constraint.
Right.
I comment it
Right.
out.
Right. Let me see the, can you show me the step bubble function?
I've declared the target volume.
Right. Right, this is all fine. Can you save equation number? Can you save the XML file?
Yes. Yeah, so after like a few MCS, like the zero would just goes to zero.
Oh, so let me check something out. I'm not sure what's happening here. 1 second. Can you go back to the step bubble
You have
function?
the cell volume defined in Python, but you don't have the Python code to do the volume, target volume. I think. There it is.
You mean like the target
So
volume?
maybe make lambda volume bigger
No,
instead of not two.
right. Mine was working, but I'm not. Yeah, make it 3. So yeah. Yeah, everything looks fine. Everything looks fine to me.
Yeah, and I think I also have troubles about like all the code you have uploaded in the folder.
Yeah.
Like in the like homework assignment like question 3, like all the things would disappear like the zero will just goes like to
Yeah, I don't.
yeah, the zero will just disappear when I run the code. I don't know why.
Yeah.
I don't know if that's my problem, like my CD3D problem, or is
Yeah, I'm
over
not
like
sure.
older.
I'm not sure.
So I'm looking at the code. So let's look at the code that you just uploaded. This is this Lambda chemo code, right?
Right.
Okay, so let me try to find it.
Let me share my screen for a second, Fambo. I'll come back to you.
Okay, sure.
But because this is what I wanted to show. The cell keeps growing as every time it hits the border, and maybe we can increase that lambda chemo a little bit. It doesn't have to, it can be a little bit higher than 5,000. But this is what I've ran this a couple of times, and what I see is that the cell velocity might decrease a little bit with the cell volume, but it doesn't decrease by much. It just basically oscillates around this this average speed, even as the cell gets bigger and bigger and bigger. That's at least what I've seen before. I was hoping that other people would be able to run it and maybe see a difference, see a little bit of differences, bigger differences in the speed. But yeah, let's go back to James. You wanted to share your code?
Give me 1 second. So
In the meantime, what about
the code the code I downloaded from the website from Canvas does not set the initial target volume and lambda volume.
right? That's what
Right.
I was. Yeah, that's the mistake I was pointing,
Right.
because I uploaded one version that has
Right.
the
So you have to
XML.
add that back.
Right.
Once it's there, it seems to work fine. And okay. So, but you do have to add that back. So I guess in Fanbo's code, we should look and make sure that he's got that added back properly.
Yeah.
Yeah, I think I specify it in the Python step file. Like
So let's take a look. Why don't you screen share and we'll take
Sure.
Because if you're having trouble with it, probably other people are having trouble. So
Right.
So in the Python step file,
yeah,
I declare the lambda and target here, and
line 28.
What?
Spelling.
Oh yeah, it's Lambda. Right. It's LAMBDA.
Okay.
There you go. That should work.
Okay. Yep.
Yep.
Okay.
Okay, cool. So at least this is what I.
Were you still looking for the file?
Who is?
Kali said.
Oh no, she's fine.
I found it. I have it.
Yep. So at least what I've consistently seen is that as the cell keeps increasing, but the speed remains relatively constant, regardless of the cell volume. So at least in the simulations that I've ran so far, the cell speed doesn't seem to change due to the cell volume. And I ran the simulation until the cell gets too big that it doesn't, the code that we have to make it go back and forth doesn't work anymore. I was wondering if someone was willing to try increasing the lambda volume instead of the target volume, and was wondering was willing to show it.
I mean, I can write it in really quickly.
Yeah.
I don't know if I have everything else updated that Fanbo does, though.
We can. It's not terribly complicated, so we can walk you through it if you want to. So yeah, the first thing is to comment out that line 30 there, because we want to specify the parameters in the Python step one. Right. And then in the Python step, I will set that line 19, which is our original chemotaxis, set it to 5,000. And that number we determined from the previous plots. And then now we have to assign a initial lambda and target volume to the cell. So inside that loop, assign a lambda volume of 50. Oh, I'm sorry, of 2, and a target volume of 50. Lambda volume of 2, and then target volume of 50. Okay, now in the step function, inside this if condition, which is where we're changing the cell's direction, in line 46, change it to be just -1, because right, that's just changing the direction. So you want to keep that because that's going to make your cell move back and forth. But then now you want to add an additional line where you increase the lambda volume of the cell. Right. You can increase it there that way, or just multiply it by 1.1. We'll see how far that gets us. Right. And then in the plot, because we were plotting lambda in line 43, we were plotting lambda versus velocity, but now we want to plot, I'm sorry, we were plotting lambda chemotaxis versus velocity, but now we want to plot lambda volume versus velocity. So that's line 43, right? Just plot, because we just have one cell, so you can just type cell that lambda volume there. Right. So now we're going to plot the speed as a function of the lambda volume, and you can run the simulation now. That should work. So right, every time the cell changes direction, then that means the lambda volume is increasing. Then we'll see how the velocity of the cell changes as a function of. You can hit if you click in window and then tile, that will format them for you. There you go. So we'll see. We gotta let it run a little bit, because depending how fast you're increasing the lambda volume, you might need to let it run for a little bit. Okay, so right.
You may want to make the x-axis linear rather than logarithmic. Or you already did that, I guess.
No, we didn't.
Or you're multiplying lambda volume by you got it. You're doing times equals for lambda volume.
Right.
I did a linear one.
That's fine. So while this runs on the background, one of the exercises, I want to explain what one of the exercises in the homework means, in case you guys want to give it a try. The last thing we wanted to test, we wanted to run this, but we also wanted to test it with the number of cells. And that is a little bit more complicated, because we know that that in Compusal, cell division happens inside of a special type of stepable called the mitosis stepable. So the way to do that is to incorporate or create a mitosis stepable inside of your function of your inside of your Python stepables. And then you have to basically copy the code that we already have and put it inside of that mitosis stepable. And then what you want to do is that you want, when the condition is met that the cell has reached the border, then you want to make that cell divide. And then probably what you would see, probably I was thinking about it, but probably the best way of doing it is to measure that, because once the cell divided, it's going to create a cluster. So and then you'll have to plot the velocity as a function of the cell number, of the number of cells in that cluster. And then probably what you need to do is you have to measure the center of mass of all of those cells, and then basically divide the cell that is closer to the border. That's the way I would do it. But there might be other ways of doing it as well. It's just a little bit complicated to implement right now, but that's the basic idea. Maybe this increase is too small because I actually don't see any changes. It's decreasing a little bit, but not terribly. I was expecting it to decrease much faster.
You might also want to try starting with a slightly larger lambda chemo so that it moves a little faster.
Right. So, right, it is decreasing. You see that it is clearly decreasing, and that was what we were expecting. As you increase the lambda volume, the cell becomes less and less motile. And one way of freezing cells in Compusal is to set their lambda volume to be huge, and that will basically freeze the cells in place. So, and you can see it here you can see it here in this plot that the velocity is decreasing as a function of the lambda volume. Right. So, but at least now you guys have an idea of what to do with in the homework when we ask you to divide the cells. And again, in that case, what you need is a mitosis steppable, and then you need to run this code inside that mitosis steppable. And the basic idea of the pseudocode is that you want to check whether the cell cluster is near the border. And then if the cell cluster is near the border, then you have one of the cells in that cluster to divide. And then you plot the velocity of the cluster, and you have to think about how to do it. Maybe take the center of mass of the cluster, and then, or the center of, you always keep track of the same cell. And
I think just tracking one cell is probably close enough.
Right. Right.
But you do want the total number of pixels in the cluster to stay approximately the same. So,
right.
So you want to divide
the cell volume.
And you have to be a little bit careful. I mean, if you want to do it quick and dirty, you divide the cells and divide the target area by 2, target volume by 2. But in fact, if you then add up the total volume, it won't be quite the same as what you would expect. And so that in principle, you should adjust the target volume to recover the original volume.
Right.
I wouldn't push it, but if what we're trying to calibrate it for real, for publication, where you were doing something more serious, you might need to do that.
Right. Right. And maybe that is something that we can come back over and next week again, and have a demo that we can walk quickly, because it will take a little bit of time to build it up right now. Okay, cool. But yeah, here the idea is that we wanted to see that as we increase lambda volume, the velocity of the cell decreases. I'm gonna take back the screen, Cal, if that's okay. Thank you. So coming back to the lecture, I'm gonna go quickly over these ideas, and we're gonna take a break, and so James can continue with the SPML integration. But as I was explaining earlier, this simple form of chemotaxis has the unintended consequence that in a field that is decreasing linearly, that has a exponential profile, the response will also be exponentially increasing. So in this particular example, I put the cell on instead of a linear gradient, I put it in a exponential gradient. And then what you can see is that the cell, as the cell moves closer and closer to the field, the speed at which the cell moves increases more and more and more, and it reaches a point that is shown in the second diagram here, that the force is so big that the cell goes poof and disappears from the simulation. So that is the computational limitation of that particular implementation. From a biological perspective, cells do not respond linearly or regimes to stimuli. They saturate their response because they cannot move, let's say, infinitely fast as the gradient increases it gets infinitely steeper. So the way to solve that is by adapting our chemotaxis, and one way of doing it is by modifying the lambda chemotaxis using the concentration of the cell at the concentration of the field at the center of mass of the cell. And what we observe in that particular case is that instead of getting a exponentially increasing response, what we get is a linear response and a saturation where the cell stops moving due to the gradient. So even though the gradient is exponential, the response or the velocity that the cell responds to, with which the cell responds to that gradient, it's linear, and that it saturates to a point where it doesn't move anymore. And if you play, I think I have a version of this code. If you play with the initial or the value of the lambda chemotaxis, you'll see that lambda chemotaxis controls how far into the gradient the cell moves before saturating and before it stops moving. And then we have this example, 9 examples that are intended to represent some biological, nice biological systems. In this self-constructed gradient example, we put a cell on a fixed gradient, on a fixed chemical field, and we had the field decay or being uptaken by the cell. And then what it does is that the cell starts moving randomly in one direction and it starts creating its own gradient. And we usually observe is that the cell has a persistent movement in one direction, and then it reaches certain points where the cell suddenly changes its direction and starts moving in a different direction. And this resembles the bacterial movement in the sense that bacteria has a combination of persistent movement in one direction and then tumbling where it switches its direction and starts moving in a different direction, and that allows the bacteria to explore its environment in search for higher areas of the available nutrients. Sorry, that scared me. And then hold on, that's making some noise.
So did you want to do that example as an in-class exercise?
I do not, because we're gonna run out of time for the SBML, and I think it's important to get there. And I'm sorry, I just got scared by that noise. So the last example is the microphage chasing the bacterium model, and we explained that this exercise, we have a bacteria that secretes a chemical field that attracts the macrophages, and they move, and then these macrophages start chasing the bacteria, and they start pushing them away. So the bacteria is the only reason the bacteria moves away from the microphage is because of at least in this particular model, is because we set it up such that there is a specific contact energy between them. And then this simulation resembles the microphage navigating in search of bacteria. And then there is a couple of questions that we can ask about this model and how it affects how adding more components to the simulation might affect the reaction of the bacteria. The reaction of or might affect the simulation. And I think we asked in the homework a couple of them, for example, adding a secretion, uptake by the macrophage, whether that would affect the general dynamics of the simulation. And we can also add a field that instead of the bacteria being pushed away just by the contact energies between bacteria and macrophages, we can add an additional field that is a chemo repulsion that makes the bacteria being able to detect and move away from the macrophage. And also, and this is in the homework as well, testing whether a saturation of the chemotaxis response as opposed to a linear response has effect on the simulation, and how the parameters must be adjusted to recover the same simulation. And those are, I think those were the 3 examples in the homework. I mean, we do not have time to explore that in detail right now. I'm willing to discuss them independently with anyone who's interested outside of the class. But for now, we'll take a break, and then James will come back. After we come back, James will take over and walk you through subcellular modeling using SPL and antimony.
I mean, many of the people in the room have been doing it already. So for you could help out and make sure we do it right. Okay. Okay. So let's say till I have 6:55 now. So let's say 7:03. We'll start back. Okay. And Josh, you said you had updated slides, or how shall we?
Yeah, I uploaded them. The latest version is uploaded in the drive. If you download it, it has all of the formats and the slides that were missing.
Okay. Thank you.
But I'll keep this up.
Josh, are you back? Are people back?
Yeah, I'm here.
So I'm trying to find, where did you put the delta notch in the cell cycle models?
They are in the drive in one
Because
second.
I was trying to make sure they were uploaded to Canvas.
I'll upload them right now.
Well, I think I've uploaded them, but you better check I uploaded the right one. Okay. So I could leave the, just, this was my Lambda chemo versus Lambda volume. I mean, the V chemo versus Lambda volume. And you see that as the Lambda volume goes up, the velocity decreases more or less linear. And in my case, when Lambda volume got to about 100, it stopped. The cell stopped moving. I don't know if other people, it's going to depend a little bit on the size of the cell and other things and the gradient, strength of the gradient, but you'll see something pretty much like that. I thought that might be useful. And it does seem that if you tell it to use 4 processors, it runs quite a bit faster. So I recommend that. If you want us to do that, we go to XML script at the top where it says number of processors. If you set it equal to 4, it should run a little bit faster. I don't know what happens if you set it to 8. I don't know how NanoHub will treat it. So for some of you, this will be a little bit of a review. For Fanbo, I guess it'll be a little bit new. So one of the things that we have, so far we've been talking about cell behaviors, and we've been talking about cell behaviors in the context of what cells do, in particular how cells move. And so the simulations we've been looking at have had cells grow, they've had cells divide, they've had cells stick to each other, we've had cells absorb and secrete chemicals and die and change type and respond to external chemical fields by chemotaxis. But we know that in reality, the cells have first, there are a lot of things we've left out. First, that the cells have very complex architecture inside the cell. The way cells move is not the kind of simple passive movement that we have. The cells have a cytoskeleton, which are a network of molecular motors of various kinds that actively move the cell membrane back and forth. They adhere to the extracellular environment and pull on it. To a limited extent, they can push as well. And we're neglecting all of that structural complexity. If we look inside a eukaryotic cell, there's also a whole variety of structure inside the cell related to synthesis of proteins and lipids and other structures. The endoplasmic reticulum and the Golgi apparat, which we've neglected. The cells have mitochondria, which are their energy functions, energy factories. Those are also encapsulated, and we've neglected them. And the cell has a nucleus, and the nucleus not only contains genetic information, it tends to be rigid relative to the cytoplasm. And so sometimes if you want to understand the mechanics of cells, modeling the nuclear compartment separately is important. All of those things are mechanical and structural things that we've neglected about the cell. The other thing that happens, of course, in a cell which is quintessential to life, is that there's metabolism. The cell takes in glucose and oxygen and produces things like ammonia, carbon dioxide as waste, lactic acid. And it also synthesizes all of the components that the cell needs to do the things that it needs to do. ATP for energy, and structural components, signaling molecules, and so on. Inside the cell, there's also signaling. We've heard about cells responding to cyclic AMP. The way cells chemotax is by having anisotropic signal coming to receptors on the surface of the cell that send information to the cytoskeleton governing the cell, the cytoskeletal polymerization, depolymerization. And so there are inside the cell networks of metabolic reactions, there are networks of signaling reactions, usually involving phosphorylation cascades. And then there also are networks called gene regulatory networks that turn genes on and off. They're responsible for things like cell differentiation. And those typically involve things like transcription factors. And so we would like to be able to model the metabolism, signaling, and gene regulation going on inside of our cells, because that's really what's controlling the cells and their behaviors. In reality, we're going to make rather simple assumptions about those regulatory signals, but we should be aware that they're there. We'll typically model those networks inside of the cells as if they were non-spatial. So we're going to treat the cell essentially as if it were a big stirred chemical reactor, like a test tube, and assume that there isn't any spatial variation in concentration within the cell. In reality, there's a lot of spatial variation of concentration within the cell, and signals often have a spatial component as well as a temporal component. So, for example, if we're talking about chemotaxis, the signal starts at the cell membrane and then modulates the polymerization, depolymerization of actin inside of the cell. If the signal is telling the cell to differentiate, the signal starts at the membrane, usually with a receptor, transmembrane receptor, and then there's a phosphorylation cascade of signal that eventually causes a transcription factor to move from the surface of the cell or the cytoplasm into the nucleus to bind to the DNA. And so signals are transduced in space as well as time. Metabolic reactions are often localized to the mitochondria or to other kinds of organelles within the cell, peroxisomes and other kinds of organelles that do degradation of chemicals. And so we're going to neglect all of that spatial heterogeneity within the cell and assume that we can treat the cell as a stirred reactor. And so in this picture I've got up on the screen, this is from a liver model that we did a few years ago. This is a model of transport where there's a multicellular model in the middle here, which is representing the flow of blood in what are called the sinusoids, essentially the capillaries in the liver. The green cells are the hepatocytes on the boundaries of the sinusoid. The blue and the red in the middle represents blood. In this case, serum in blue and red blood cells in red that are moving down that sinusoid. And they're exchanging oxygen and other chemicals with the cells around them. And in this case, we actually have an explicit model of the fluid flow, the kinds of things that Jeffrey's doing in more detail now. Within those green cells, in this case, the cells are modeled as absorbing acetaminophen, Tylenol, and acetaminophen in turn is modified by enzymes within the cell, in particular a series of enzymes called cytochrome P450s, CYPs. And these cytochromes make certain molecular transformations to the cell, to the molecule. CYPs are primarily responsible for the breakdown of molecular species. Some of that breakdown is to make nutrients available. So breakdown of proteins, for example, to make them available, making glucose into glycogen for storage, and then putting it back as glucose and releasing it from the liver. But one of the main things that the cytochrome P450s do, the CYPs do, is that they break down toxic chemicals, or at least chemicals that the body thinks are toxic, into things that the body thinks are less toxic. In particular, some of these cytochromes do what's called glutathione conjugation, and you probably need to be a better biochemist than I am to go through all the different transformations that cytochromes do. They can add sulfates, they can add all sorts of different decorators to the basic molecule, and each one of the cytochromes works on particular organic molecules and modifies it in a particular way, and there are a lot of them. In the particular case of acetaminophen, unfortunately, one of the things that happens is that one of the cytochromes that normally is important for the breakdown of molecules to detoxify them, and this also happens with ethanol, which is how you get hangovers, is that it actually makes it into a toxic byproduct. And so while acetaminophen itself, APAP, is not relatively non-toxic, one of its breakdown products, called NAPQI, is very toxic. And it is toxic because it scavenges glutathione, which is a necessary antioxidant in the cell, irreversibly. And NAPQ binds then to glutathione and takes glutathione out of circulation. And if cells have too much glutathione disappear, they die. And so too much Tylenol can lead to the death of hepatocytes in your liver and potentially the liver fat. And so this was a model where we explicitly modeled acetaminophen being transported in the blood, being exchanged with hepatocytes, and then being metabolized. And so in the model of this kind, we want to be able to measure the supply of glutathione, of acetaminophen to the cells. And then within the cells, we want to have an explicit model of the chemical reactions, in this case, metabolic reactions, that are leading to depletion of glutathione, formation of NAPQI. This model also was embedded in another network model, in this case, a network model not of metabolism, but rather of transport. When you eat food, the food goes into your gut, into your stomach, and then into your intestines. And different components of the food are broken down by the acids in your gut and by the bacteria in your gut and absorbed in various places along the intestinal tree. Once the breakdown products are absorbed, they're distributed in the blood to the various organs. They're further modified in the liver. Some of them are broken down and eliminated in the liver. They're also modified and eliminated in the kidney. And so there's a transport model in this case, which is 1B here, which represents the flow of blood in the body, its distribution to the various organs, and the elimination of some molecular species in the form of a bile or urine going into feces. And in some cases, also some molecules are exhaled or sweated out in other ways, although they're usually less significant. So if you're going to build a model of the whole body and how, say, a drug moves around in the body, in and out of a tissue, you have to have a non-spatial model, a network model of what's called ADME, absorption, dosimetry, metabolism, and excretion at the top, and inside the cells, a model of what the signal is doing, what the molecules are doing in terms of metabolism. We could also have models of, again, of signaling and gene regulation. And so all of these top level models and these bottom level models have the form of networks, of dynamic networks. And Compusal, as we've seen already, Collie's talked about it, and Tyler's talked about it to some extent. Compusal allows you to build simulations in which you run network models either inside of individual cells. If we ever get to compartmental cells, you could run them inside different compartments if you want to have different subcompartments. And also potentially in the background, so what are called free-floating models, which live in the whole system. And this is a model that I think we may or may not get to run today, but I hope we get to. If we don't do it today, we'll do it next week, is a model of cell cycle. In our very simple work so far, we've assumed that the cell grows at a constant rate that divides when it gets to a target volume. In reality, there are a whole series of molecular species, in particular the cyclins, which oscillate inside of cells that are going through cell cycle. And the relative concentrations of the various cyclins determine how the cell enters and leaves the various phases of the cell cycle. And in this particular case, this is showing a movie, rather old movie, because you can see the colors don't line up with the boundaries of the cells very well, of a model of a cell cycle that was developed by John Tyson at Virginia Tech, which has been loaded into each cell individually and is running inside of each cell, determining how that cell grows and divides. In this particular case, one of the things that's interesting about that model is that one of the ways the cell cycle is driven is that as the cell grows, certain chemicals that are not made all the time during the cell cycle are diluted by the cell growth. And it's actually the reduction in concentration of those particular molecules that drives parts of the cell cycle. And that's why when you do the simulation, you see that not all the cells are in the same phase of the cell cycle. As the cell grows, there's a stochasticity to the growth that leads to a stochastic behavior of the cell cycle model. Initially, they're in synchrony, and then as time goes on, you'll see them lose synchronization. This kind of biochemical kinetics shows up a lot in oscillations. People love modeling biochemical oscillators because they have very distinctive patterns. And of course, the last semester, Jeffrey was modeling neuronal oscillations. But there are a lot of situations in which oscillations aren't what you need or want, or the most functional aspect. And there are other kinds of network that are quite important as well. Ironically, a lot of molecular species oscillate not because the oscillations are important, but precisely because they're not important. If you build very complicated networks with a lot of negative feedback, it winds up that they tend to oscillate. And in many cases, that oscillation doesn't make any difference. It doesn't do any harm. And so the system tolerates it. Whether that oscillation is functional or not isn't necessarily very well understood. And we'll talk about a particular one today, which is delta notch patterning, which is not primarily an oscillatory pattern, at least in time. It leads to spatial oscillations rather than temporal oscillations. And delta notch is very important in development. In particular, vascular development is predicated on delta notch patterning. This is a delta notch patterning mechanism. And then this is the one I hope we'll be able to run today before we break. In this case, you have two molecular species, delta and notch, and we'll come back to this in more detail in a minute. with the property that it's a little bit, if we were a physics audience, we talk about magnetism. We call ferro and ferrimagnets. In this case, delta cells, high delta cells like to be next to high notch cells, and vice versa. But high delta cells don't particularly like to be next to other high delta cells, and high notch cells don't like particularly being next to high notch cells. And so you have something that's trying to build a checkerboard. We've seen checkerboards due to cell adhesion. This is a molecularly induced checkerboard, where you spontaneously get this kind of patterning, where you'll see the high delta next to the high notch and vice versa. And that's a simulation we'll run. In this case, the signaling is done by contact between the cells, and we'll think about how to implement that as well. So in one case, in the first case, in our cell cycle model, the cells weren't signaling to each other. The oscillations were controlled by the volume of each cell independently, even though there was a complicated oscillator running inside of each cell. In this case, the chemical reaction or the signaling network within each cell is quite simple, but because each cell is exchanging information with its neighbors, the overall patterning is fairly complex. And then we have more complicated models which mix these things together. This is a movie we've showed at the very beginning of the class. This is a model of segmentation of the formation of the pattern that's going to lead to the vertebra. In this case, we have a gradient of a morphogen, in this case FGF8, fibroblast growth factor 8, which is high at the tail of the animal as the animal is growing and decreases as you go towards the head. FGF8 maintains the cells in an undifferentiated state or a relatively undifferentiated state. In addition, within each cell, there are a series of oscillating genes. In mammals and in birds, there are 3 different independent oscillators, each one with slightly different properties and each one of which couples. In fish, there's only one that do the same thing. And what seems to happen as the tissue grows, you'll see on the right-hand column, you'll see these waves, apparently waves. They're actually not waves. They're what are called pseudo waves. These temporal oscillations, which also lead right to apparently spatially traveling waves of gene expression. And when the cells cross a particular threshold level of this middle column, FGF8, they read the phase of the oscillator inside the cell, and then they differentiate to change their adhesivity according to the phase of that oscillation. And so in this case, there's also delta notch, as it happens. N stands for notch. There's also contact signaling among the cells, which is how they synchronize. And so in this case, this simulation, we have an external chemical field that's modulating the cell behavior. We have an internal cellular oscillator, and we have synchronization of the oscillator by contact. So there are a whole variety of different kinds of information flow in this system, but ultimately it comes down to a network of signaling and gene regulation inside of each cell. And so we want to be able to add these kinds of dynamic networks into Compusal. And there are 2 basic language ways to do it. There's a language called System Biology Markup Language, SPML, which is an XML, like the Compusal 3D ML. And SPML is a language for writing down networks of reactions and rates. Now, the one disadvantage of using SPML is very widely used, but the one disadvantage of SPMLs is not designed to be read by people. It's very verbose and very repetitive. And so people don't typically read and write SPML directly. They use a model editor that either represents the SPML graphically in the form of a network of nodes and arrows and rates, or uses a different language to encapsulate the information in the SPML. And we've used Tellurium last semester, and we'll use it again as a way of writing models. And those can then be turned into SPML, and Compusal lets you import SPML models into cells or into the whole model. The advantage of being able to read and write SPML is that SPML, there are a lot of models that have been published using SPML. And there's a biomodels database, which has hundreds of published models. And that means that if there's a published model of gene regulation or signaling or metabolism that you want to use, you don't have to write it from scratch. You don't have to retype it. You can pull it in directly from the biomodels database. You have to know what the units are, so you can convert the units and connect it to your Compusal cells, but you don't have to rewrite it. The other thing which we developed a little bit more recently, which is what we'll use mostly, is that the language antimony, which is a nice text-based language, a very simple way to describe chemical reactions inside of cells, can be edited using Twidit. It's designed to be read by human beings. And so you have the option of importing SPML or antimony, or typing antimony in Twidit. And again, those can be run inside individual cells or in the whole simulations. Once you've loaded your network models into this whole simulation or into the cells individually, they're converted into internal representations and then solved by a solver library, which Andy Sumudji developed, and run inside of Compusal. Just a word or two about SPML. SPML is in software. It's a standard for describing networks. And it's really what's called an exchange language. It's like PostScript for printing. It's not something that humans are ever supposed to read. It's something for computers to talk to each other. But there is this biomodels.net, sysbio.org. There are many repositories of these kinds of models. If the standard thing that we're going to represent in any of these situations, and the whole class last semester was about it, I'm not going to go through that again, is chemical reactions. Here is a simple chemical reaction where a species S1 is breaking down to become 2 copies of species S2 at a rate K. You have to specify how much of S1 and S2 you have at the beginning. You have the choice of whether these represent concentrations or amounts. And then you have a rate constant of the breakdown, for example. And that would be a typical rate equation. And this is what that very simple one-line rate equation looks like when you put it in SPML. And it's not that SPML is impossible to read. It's just verbose and slightly awkward. Says that it's XML version 1. There's some things about the particular kind subversions that you've got. SPML, because it's designed to work with those pharmacokinetic models that I showed, has a concept of compartments and the volume of a compartment. So if you want to say that certain things are happening in the liver that has a volume of 2 liters, and some things that are happening in the blood, which has a volume of 7 liters, and other things are happening in the lungs that have a different volume, you can specify how big the compartments are. you have to declare your species. That'll be familiar from Compusal, where we declare the cell types. And you'd say whether these are variables or fixed boundary conditions, initial concentrations, which compartment they live in. You have a list of parameters, and then you have a list of chemical reactions. In this case, there's only one chemical reaction. And then you have to write the mathematical form of the rate equation. And this uses a language called MathML that's extremely verbose, but is something that computers are good at parsing. And so this one line diagram here turns into these lines of code. Again, if we walk through it, we can see exactly how each line corresponds, but it's not something we want to read and write by hand. What we'll use instead is antimony. For people who haven't worked with antimony before, it's very nice if you search. Josh, could you put the link to the antimony documentation into the chat for Banvo? If not, I can paste it in. It's really a very simple modeling language. Again, we're typically describing chemical reactions in the form of rates. It handles things like stoichiometry. If 2 molecules of A combine with 3 molecules of B to make 7 molecules of C, it handles all of that conversion automatically. But the basic form is that we have a model string. We just have literally an alphanumeric string. Double backslash represents comments in antimony. We can name rate equations. And so here we have rate equation E0. It says boundary species S becomes R with a rate K1 times S. So the rate at which we produce R is K1 times the concentration of S. R equation 2, E2, 1 becomes R goes to Q at a rate K2 times R. Q goes to nothing, that's to K, at a rate K3 times Q. And I can write any simple algebraic form, and their basic mathematical operators, mathematical functions like sine, cosine, logarithm are all implemented. The only thing that you're likely to run into in antimony that's a little more complicated is the concept of an event. You can have if statements inside of antimony that work in a slightly funny way. The other thing that you have to watch out for in antimony, which we ran into it earlier today, is that antimony is what's called the declarative language. So it's not code that's executed. It's a specification of what code should be executed. And that means that the model that is written here is read once and turned into internal computer representation. And in particular, equal signs are only evaluated at the very beginning when the model is parsed. And so rates are dynamic, so they're evaluated all the time, updated all the time. But equal signs are not. Their declarations are only evaluated once. And so there's a special kind of equal sign called colon equals that tells you that it's really a rate, and so that it has to be updated repeatedly. And we're not going to run into that today, but it's something to be aware of. We can come back to it if you need help with it. So we create a model string that specifies what the chemical reactions are. And an example of this might be, here is some, we're not actually going to do this one, but here's some example of some complicated signaling network. An activator from outside comes in, it biases a kinase to phosphorylated state of the kinase. That kinase phosphorylates another kinase to its phosphorylated state. That phosphorylated molecule acts as a inhibitory transcription factor for the production of protein E2, It acts as a positive transcription factor for the production of protein E3. E3 promotes the degradation of S. E2 also promotes the degradation of S, and S is synthesized. So this would be an example where we have a metabolic network, which is the destruction of S and the synthesis of S. We have a gene regulatory network through E2 and E3, and we have a signaling network here on the left. So this was just a schematic of the possible ways that a network can represent these three fundamental biological concepts of metabolism, regulation, and signaling. So what we might do is we might take this network and we might load it into each cell individually in our Compusal simulation. Or we might load it into one cell and not into other ones. And we might also imagine loading it into, say, only the blue cells and not into the green cells, because this network might be active in one cell type and not into others. Or you might imagine that this network actually is running not in these cells, but in some cells in the background. And so you might load it into the background, what's called a free-floating model. And here on this slide, we have essentially all of the functions. These are all available if you go into the Twit and go to Spml. Let's see where, let's go into Twit. Here, let's go into, we're not using it here in this model, but if I go into Twit, and let me just show you what the options look like. I go up to CC3D Python, I go down to Spml solver, and you'll see there are a lot of options. There's model initial data, there's add antimony model to cell, and then there are a bunch of options. You can add it to cells of a particular ID. You can add it to cells, all cells of a given type. You can add it just to one cell, whatever that cell is. And there are other ways, there are other standards for describing network models. There's a standard called, one of the things that they joke is that one of the nice things about standards is there's so many to choose from. Of course, if you have two standards, then they're not standard, in the sense you have to convert them. But there's another language called CellML, which is used by some people. And you can read and write CellML. And pretty soon there's going to be yet another one that we're going to support as well. But there are multiple standards for this kind of specification. Or you can add SPML models to cells. All of these do the same thing. And then there are those free-floating options. So you can either attach the model to a cell, or you can attach the model to the simulation as a whole. And the key things that you see there also would be time-step SPML models. You have to take the integration of forward in time for the networks. And you might want to be able to clone SPML models from one cell to another. You might want to be able to delete them. When cells differentiate, some component of the system disappears. You might want to be able to delete them. And then there are various ways of setting the particular options for the integrators and things like that. The main ones that we're going to work with here are how to define a model, and we'll be using antimony primarily. So we'll say string equals, and then you put the antimony model in the string using triple quotation marks so that carriage returns are allowed. And then if we want to add an antimony model to a cell, we say self.add antimony to cell. We give the model string identity. We give it a name because we're going to have a way of referencing that model, because potentially there could be multiple models loaded in each cell. In this case, we're adding it to a particular cell, and we can set the time units of of the antimony simulation relative to the Monte Carlo steps if we like, so step size. It's also possible to add overwrite initial conditions and do some other more complicated things. The syntax is essentially parallel, whether we add it to a single cell, to a cell specified by the cell ID, to all cells of a given type, or to the model as a whole. That's done usually in the start function. When cells divide, these models are copied automatically when we do a clone, which is very convenient. And then in the step function, we'll have a command which is self.timestep SPML, open close parentheses. And that will automatically time step all of these models in sync with each other. And that's really very convenient because it means that we don't have to do very much. We can load, if we have 1,000 cells, we can load them all with a model of some signaling or metabolic network with a single line of code, and we can have those models execute with a single line of code. If we want to know what the value of a particular variable in our SPML model or antimony model is, we have to remember the name of the model from where we defined it. And as usual, everything in our world are dictionaries. So cell.spml.model name, square bracket, quote, and then the name of the variable or the parameter in the antimony. So if in the antimony model we had a variable capital A, and the antimony model, well here, it was just literally model name, cell.spml model name, quote, capital A, will return the value of A in the internal model. So we don't have to do anything to take information back and forth between the subcellular model and the Compusal. It's available in the form of a dictionary. The one thing we might need to do is if we want to use a tracking field, we have to copy the value into the tracking field. And to read and write these variables, we don't have to do anything. We say variable equals cell.spml model name. And if we want to update it, we can overwrite it cell.spml model name, et cetera, equals value. That's really all there is to it. Copying models and deleting models isn't something we're going to do that often, but they're available. And so basically we need to know how to create a model in the first place, string equals antimony, how to load it into a cell, self.add antimony to cell, how to execute it, self.timestep, and the fact that to read and write the variables and parameters, we use dictionaries. And so there are 4 or 5 commands that we have to know. And so I guess we're going to run out of time. But in delta notch patterning is a critical kind of, as I mentioned, this is what's called the juxtacrine signal. It cells communicating by contact, and it's used again and again and again in development and in adults to maintain tissues. In this case, the photoreceptors in the eye of a zebrafish, an embryo, a fish embryo. Here, the hair cells in Drosophila in the ear. Here, the cells in the intestinal crypt. In each one of these cases, you have this pattern where you have isolated low notch cells surrounded by high notch cells. At the molecular level, this works by transmembrane signaling. You'll have actually on both sides delta and notch in the membrane. When delta and notch bind to each other, here it starts here on the right in this picture. Here's notch in one cell and delta in the other cell. When notch binds to delta, there's a conformational change that means that the notch is cleaved at the extracellular side. And so you're left with delta bound to a piece of notch on one side. That sends a signal on the delta side. And the intracellular part of the notch, what's called NICD, notch intracellular domain, comes off the cell membrane and goes inside of the cell, where it acts as a transcription factor. And so this is really something you see again and again and again. And so this is a signaling network. It's a loop, but it's a loop that you can't define in terms of one cell, because the notch in one cell signals to the delta on the neighboring cell. The delta on the notch on the neighboring cell signals to the delta on your cell. And it's a cross inhibition loop. Ultimately, one or either notch or delta wins in each cell, and you wind up with a situation like this, where you have high delta surrounded by low delta, high delta surrounded by low delta. So it's a little bit of social distancing. The delta cells move to isolate themselves from each other. And this kind of patterning is very typical in development. It's typical also in wound healing, and it's in vascular development, and it's messed up in cancer. There are almost an infinite number of delta notch models. Some of them can be very complicated. Some of them are not so complicated. The simplest delta notch model that I know of is this Collier model from 1996, which doesn't explicitly model the difference between delta and notch signaling on the surface of the cell and inside the cell. It's going to say that each cell has a number which represents its delta level. Each cell has a number which represents its notch level, which is a little bit different from what's happening in reality, where you have lots of individual receptors on the surface. And there's a feedback where within each cell, high notch turns off delta. You see that here. The change of delta goes inverse with the amount of notch. Delta decays by itself. So if I had no notch, if I have no notch, I would have delta dt equals nu times 1 over 1, constant source, minus delta. So delta would go to 1. The level of delta would go to 1. As the notch goes up, the level of delta goes down. Notch, in turn, is activated not by the level of delta inside the cell, but by the level of delta on the neighboring cells. So this delta average here is not the amount of delta inside my cell. I don't care. It's the amount of delta I'm getting from my neighbors. And in this case, higher delta on my neighbors causes higher notch for myself, and there's a decay term. You have that? So again, you can't really understand this when looking at a single cell. You have to look at two cells at once. If we run that, if a cell has a high, if the cell is surrounded by high delta cells, it downregulates notch. And then we'll see a picture like this where you get high notch cells separated by low notch cells. Yeah. Okay. And so what we're going to do is, and we're not going to have time to do it today, so we'll start next time. Maybe you may get this as a homework assignment, depending on how far we get. Is we'll fill the space with cells. You've seen the results of the simulation already. We'll fill the space with cells. We'll put this model in using atomony. It's only a few lines. We'll add it to each cell. And each cell, we're going to loop over all the cells. Then we'll look over each neighbors. We'll see how much delta our neighbors are sending to us. We'll use that new average neighbor delta to evaluate this dN by dt term, and we'll visualize the pattern. And this is what you get when you run that. And this is visualizing Notch. And what you see here again is that Notch, you get these continuous background of high Notch and low Notch values in this checkerboard pattern, isolated, more of a leopard spot pattern than checkerboard. And so we're not going to have time today, we're out of time. But the next thing we'll do, again, this may turn into a homework problem, will be to implement that little simulation and explore how the geometry works. If you have a one-dimensional array, linear array, each cell has 2 neighbors. If you're in a square array, each cell has 4 neighbors. If you're in a hexagonal array, each cell has 6 neighbors. Each one of those leads to slightly different delta notch patterns. Okay, so I think I'll stop there. The other exercise we'll do is going to be doing delta notch patterning with growth and cell division. It happens that delta and notch both influence cell cycle. And we'll see that as the tissue grows, the pattern will evolve. And so it's actually rather an interesting exercise to see how they interact. So sorry we didn't get to do that exercise tonight, but I thought it's important to give some background on these ideas. Is there anything people want to talk about before we break? I'm sure people are tired and hungry, but you're welcome. Any questions about this before we break for the evening? No. Again, Tyler and Kali are already using this, so in a sense, this is review for them. Jeffrey you've seen these antimony models last semester, but not in the context of Compusal. And Fanbo, probably these may be newer for you. Okay, well, thank you, everybody. I'll see you again next week. Happy to meet with people to talk about their individual projects and help debug ideas. So I'll thank you again, and I'll see you in a week if I don't talk to you before.